Right. Well, thank you, Faith. Okay, there we go. Yeah, certainly. Thank you for the invitation, Faith. And as Faith mentioned, I'm Tucker Patterson. I'm the Deputy Director for Research at the National Center for Toxicological Research, which we call NCTR for short. And as you can see why most people cannot pronounce toxicological with the first try. I'm going to talk a little bit about NCTR and the research that we're doing there, and also that we're pretty much the best kept secret in Arkansas. If you don't know about NCTR or where it's located, we indeed are in Arkansas. If you take I-530 from Little Rock to Pine Bluff and you pass exit 24, you'll see NCTR, its own big blue sign there at the exit. And we have a summer student program that we've had many students from both UCA and Ouachita participate in over the years. And I'll talk a little bit about that towards the end of my presentation. But I want to give you a very high level presentation here. I want to touch a little bit about all the different types of research that we're doing at NCTR, because as you know, in chemistry is a very broad area, and it really branches into most every scientific field now. And I'm going to tell you a little bit about some of the work that we're doing there. Let me go ahead and stop my video here. Okay. So this is a picture of our campus. So we have about 550 acres there in the piney woods of Arkansas. And we have a little over 30 buildings, about 1.2 million square feet of laboratory space at the center. And I will point out that we always have to put this, because we are a regulatory agency, that this presentation does not necessarily reflect the views of the Food and Drug Administration, but they are my views of NCTR and the FDA. So we are a unique FDA resource. There are multiple centers within the FDA. We are one of 7 centers across the agency. All the other centers are located in the DC area, specifically in Silver Spring, Maryland, and the Rockville, Maryland area. But we were established here in Arkansas in 1971 as a non-regulatory national research. We are owned and operated by the agency. We do toxicological research, and we collaborate with other government entities, as well as industry and also in academia. And we try to satisfy the mission of the FDA, which is to promote public health. This is our organizational structure. Our director, William Slicker, is probably our longest serving director now, over 15 years as our director. He came to NCTR in 1978, I believe, and thought he would stay a couple of years. And so he stayed a little bit longer than that. And so he's working on year 43 now, and he's our current director. And you can see the upper part there is more of the management aspect. I actually reside in the Office of Research. I oversee these 6 research divisions, and you can see the various types of research that we do, just descriptive there in the name of the division. This is our current staff. We have about 700 employees on campus, but we have nearly 300 FTEs now, or what we call full-time equivalents. These are permanent government positions. Out of that staff, about almost 150 research scientists, and then our support scientists that would have, bachelor's degrees, master's degrees, doing our lab support, and then a little less than 100 on the administrative side. Our contract staff takes care of our animals for us. We have an animal care staff. We have a pathology contract on site, maintenance staff as well. And then our ORISE, that's our postdoctoral fellows, and we typically run about 50 postdoctoral fellows at any one time. And out of those 50, usually anywhere from 30 to 40 countries are represented in that number. These are our primary research goals. We want to advance scientific knowledge and the tools required to support personal, animal, and public health. A lot of people don't realize that the FDA, especially through the Center for Veterinary Medicine, we regulate any type of products for animals. And so your household pets, your livestock, all of those Medications are approved, and of course, one of those that's been in the news quite a bit lately is ivermectin, which is a anti-parasitic that's primarily used in livestock, and so that's got a lot of press lately with the COVID pandemic. We do want to enhance collaborations with our other centers, and we do promote global interactions. We have several groups that are involved in the World Health Organization, that are involved in the Global Summit for Regulatory Science. And so, we want regulatory science to be promoted across the globe, and we want the types of stringent criteria that we follow within the agency to be followed across the globe now, especially considering all the imports that we have with our drug products and also the constituents to make the drugs. Just a little bit about our portfolio. I mean, we have a very wide variety of things that we're working on, but you can just see some of it here. I'm not going to read every one of these, but I'll go into a little bit more in depth into some of these as we get through the presentation. Of course, our emerging technologies, that's a big area now, looking at metabolomics, lipidomics, proteomics, and bioimaging. A lot of computational modeling now, looking at big data. That's a huge emphasis now within the agency. And I'll talk a little bit about our alternative models, like our organotypic and stem cell systems. And again, these global outreach areas that we're heavily involved in now, and international standards, methodologies, we're heavily involved in. So looking at our just support across the agency, this kind of gives you an idea that of our collaborative portfolio. We try to collaborate with the other FDA centers, but also again with academia, and industry. And you can see that the drug side, CDER is a Center for Drugs Evaluation and Research, and of course they're going to approve all your pharmaceuticals that you're taking. Then you've got the Center for Biologics here. Of course, they oversee our vaccines. We don't do as much work in that area, but we do some. Here's the Center for Veterinary Medicine, the Center for Devices and Radiological Health. So any device that's put in your body is approved by the FDA. So this would be, hip replacements, knee replacements, stents, any of those types of devices, pacemakers. They're all going to have to be approved by the agency. So at any one time, we're going to have 300 to 400 ongoing research projects. You can see this was done back in July. I did this presentation for an FDA 101 seminar that I was doing, but we had about 340 ongoing projects at the time. Again, almost 60% of that is conducted in collaboration with the other FDA centers or their Office of Regulatory Affairs. Then again, 22 academic institutions, federal agencies, and we also have a memorandum of understanding with the state of Arkansas. But over here on the right-hand side, you see just the things that we're involved in that we want to have an emphasis on. And I am going to highlight some of these issues with COVID-19. We were able to start this very early in the process. This was back in February of last year when the pandemic, we saw that it had reached the U.S. and so we started gearing up for studies in that area. And so we started 21 studies at the time, and we've got about 10 more now that are in development. We had special COVID funding. It seems like everybody had COVID funding there for a while, but these are some of the projects that we were able to fund using this earmarked coronavirus funding. And then we had other funding from our Center for Drugs that funded a couple of more projects that I'll go into a little bit more detail about later. So this is one of the projects that we were doing with our coronavirus funding. This is a recombinant spike protein study that we were doing. And so the aim of this study was to produce reagents to support the COVID-19 research. And we were investigating if other coronaviruses or if antibodies to the spike protein of SARS-CoV-2, if they could possibly cause antibody-dependent enhancement of the disease in the presence of SARS-CoV-2 or other human coronaviruses. So the figures show the purified spike protein of the NL63 and the SARS-CoV-2 that was in the form of monomers and trimers, and these fluorescent cells that you see, they're actually infected SF9 cells that are expressing the SARS-CoV-2 spike protein that's conjugated with GFP. So these are some of the publications that we already have in print in the COVID-19 space. And so, I can send these out if people are interested in that, but I have the DOIs there. And so they're easy to be, they're all hyperlinked there. And so you can just click on that and it'll pull that up and you can go right to those publications. I do want to mention the National Toxicology Program because this is a partnership that we have. It's through NIH, the CDC, and also the FDA. And the primary players in that for NIH are the National Institute of Environmental Health, and then also NIOSH that looks at, of course, worker safety. And then NCTR is the primary player for the agency. But this partnership was established in 1978. And what we do with this partnership is we look at the toxicity of chemicals and other substances of public health concern. And, we want to strengthen the knowledge base in toxicology, through this agreement. So this is an interagency agreement between these entities. And the IAA was actually established in 1992 to conduct toxicology and these mechanistic studies at NCTR. So the way this works, if we want to nominate a compound to examine, and the other groups within the NTP, which would be NIOSH and NIHS, if they agree, then this work is going to be primarily done at NCTR. And so you can see that we have some ongoing studies now on inorganic arsenic, and that's with the Center for Food Safety and Applied Nutrition. We're looking at polyethylene glycol, and that's primarily with the Center for Drugs and the Center for Biologics. We use an in vitro air-liquid interface model, and I'll talk about that a little bit more in detail lately. And of course, the microbiome is big in the microbiology area. And so now we're entering a new phase, and we're more focused on these direct collaborative efforts between the groups to address these strategic interests of the NTP. So these are just some of the compounds and some of the agents that we've looked at over the years, and you can see that it really runs the full gamut of different compounds that have been examined over the years. And of course, bisphenol A just a few years ago was in the news, and there's been numerous studies done, and those were primarily for the government, were run out of NCTR. So one of the capabilities that we have now is an inhalation facility, and we do this primarily in conjunction with the Center for Tobacco Products, which is the newest center within the agency. Of course, just a few years ago, Congress mandated the FDA to start regulating cigarettes and tobacco and those constituents. And so, of course, the primary toxicity from tobacco is going to be smoking. And so we developed this inhalation toxicology area to look at these chemicals that are contained in tobacco, or they're formed during the tobacco combustion process. And so we can do both in vivo and in vitro. And in the first compound that was examined was NNK, which is a known carcinogen that's produced in tobacco smoke. And those studies have completed, and we have publications on that. And now we're looking at actually nicotine, both in vivo and in vitro. And this apparatus on the far right is an in vivo system. And then I'll show you what our in vitro system looks like in just a few minutes. And so another one is the alternative microfluidic systems, microphysiological systems. We're doing a lot of work in that area now. So a variety of models are being used. We're looking at neural stem cells, cardiomyocytes, bronchial epithelial cells, germ cells, testicular cells, pancreatic organoids. And so really we're covering the whole body and the whole physiological system through these different methods. One of the newest things that we're looking at are these brain chips. And this is a collaboration, again, with the Center for Food Safety and Applied Nutrition, also the Center for Devices and Radiological Health, and the company Emulate. And Emulate has created these organ-on-a-chips, and in this case, the brain chips. And they have an integrated neurovascular brain chip that they see similar characteristics as what's observed in Alzheimer's disease. And so we're setting this system up now in our neurotoxicology labs at NCTR, and that's within our division of neurotoxicology, and starting to try to validate these systems and compare it to some of the known in vivo things that we have some of that data. And so we can hopefully mimic the Alzheimer's brain using this chip, and we can use this as a drug screening platform prior to it going into an in vivo model. So here's the human brain chip, and you can see that it mimics all the different cells within the brain, the neurons, the microglia, the astrocytes. And so those are all representative. And this is the pictures on the right show successful chip establishment. And the top image there is a GFAP that's for astrocytes. And the bottom image there, that's labeled with occludin for the endothelial cells. And so we now successfully established those chips. And so we're moving forward into actually doing experiments now. So this is another a different model that we're using. This is a microfluidic system luminal structure for the in vitro mouse spermatogenesis. And so we're looking at the evaluation of reproductive toxicity. That's, of course, an important component of safety assessments in drugs. You want to see, how that's going to affect, reproduction. And I'll get into a little bit more on that later, and some of the things that we're doing with some of the different virus models that we have and their impact on reproduction. And so in this establishment of this particular model, we're doing a comparison to conventional tests. Conventional tests are very labor intensive. They use large number of animals. And so we're trying to use these models to, of course, satisfy the 3 R's, where we reduce, the animal numbers that we're using now when we do test these agents. And so we're trying to develop a 3D testicular model, and we're going to link this to a 3D liver model that we've already established. And using this in vitro testicular model, we can evaluate product safety, and hopefully that will complement our animal studies by providing information for study design and what agents that we can prioritize and first look at in our animal models. And also dose selection is a very important part, where you're not using a large number of animals just to try to find, the proper dose to use. And so these models also provide information for interpreting testicular toxicity data that have been already derived from animal studies. And this is the way the microphysiological system looks like when you're doing a testicular model. And this particular model, we're looking at the Zika virus. And we all know that if you've done any research into Zika, the biggest issue there is when women are infected with Zika while they're pregnant. When they have offspring, a lot of times the children are inflicted with microcephaly. So they get this hydrocephalus and just a larger brain, kind of the fluid on the brain issue. And so we've developed this model that's basically a 3D model looking at the Zika virus, where we can see if it crosses over. The human placenta, of course, transplacental transfer of agents is a very, very important issue. You see there that nearly 90% of women use at least one medication during pregnancy. So, any maternally administered drug may be toxic to the placental barrier and impair its ability. So, you want to look at that. Is it going to affect the fetus? Is it going to cross? If it's causing, if it's supposed to be causing protection for the mom, or it's going to be a therapeutic for the mom, is that the proper dose for mom, that she still gets a therapeutic effect without harming the fetus? And so we're looking at these experimental models, currently mouse models and human placental lines. They have limitations in recapitulating the structure and function. And so, we're looking at these different systems where hopefully we can test these agents, more in an in vitro system that mimic the human placenta. And we also have this human placental immunity model here on the right, and that's comparing to the human in vitro system and also the humanized mouse model, where we can evaluate utility and reliability of these barrier models. The air-liquid interface human airway tissue model, we first started using this with our studies with CTP. And this is a really fascinating model because although it's an in vitro model, it completely mimics the human airway. And so you get the architecture there, just like the human airway, where you get the differentiation of all of these different cells. And so one of the things that we first did with this human airway model, we actually have instruments that will physically smoke a cigarette, and they'll blow the smoke across this in vitro model. And you can see that the ciliated cells, the little hair-like structures here, that they would actually flow as the smoke, flowed across there. You could see them, moving, and then we can test this in vitro system to see if there's any gene expression issues with the tobacco smoke and its effect on these cells in vitro. And so they differentiate again to include all the different types of cells found in the human airway. And so this is the mucus secreting cells, the goblet cells, and the beating ciliated cells. So this is what this model looks like. And we can look at the effects on the human cells by looking at this in vitro system. So one of the applications that we're using now, of course, with COVID is the issue with the human airway, and the ACE2 receptors there have shown to be affected. So this is a study that we started, and we're hoping that this is going to provide better understanding of the effects of the ACE2 modulating drugs on SARS-CoV-2, both its entry and infectivity using this model. Our nanotechnology core facility, or we call it the nanocore for short, that has a lot of capability as well. We have transmission electron microscopes, scanning electron microscopes, and so we're looking at research on the detection, identification, characterization, safety, biocompatibility, and also standards developed is a big issue now, and we're looking at that and developing standards. Really, these are global standards now that are coming out of our nanocore. You can see that this helps with regulatory review. FDA has found that has these formal standards recognition program to identify standards that can aid in regulatory review. Several other FDA identified items are being developed as standards from the nanocore, and you can see that we have recently a consensus to bring 2 international standards through the ASTM to bear. And we have also 3 more standards on lipid composition that are in the final stages of approval. Microbiology, of course, our division of microbiology is another focus area. We have a lot of capabilities there. We want to evaluate the impact of the antimicrobial agents. Of course, food contaminants is an issue, of course, with recalls, and we all hear about the salmonella issues or E. coli issues. So also food additives are something that we look at. Nanomaterials, sometimes that's in packaging. In food packaging, does it cross from the packaging over into the food supply and then get into the human system as we consume this? So we're looking at methods to detect and characterize microbial contaminants in FDA regulated products. I didn't mention that, right now, the FDA is responsible for regulating about 20 cents on the dollar when you start looking at, the food items we regulate, the drugs, the biologics, the veterinary medicines, the tobacco products. And so we have a pretty heavy lift there for our agency. And people think about the FDA maybe being this, large government agency, but we actually have a little under 20,000 employees now worldwide, which is actually less than UAMS employees. Our medical school there in Little Rock employs over 20,000. So we are smaller than UAMS, and we're regulating 20 cents on the dollar. Of course, one of the things that the Division of Microbiology has been doing is of course, looking at women's health and tobacco products and nanotechnology as well, trying to mesh all of the different aspects that we're doing at NCTR together. So we do have a lot of crosstalk at the center. One of the things that's always interesting, and most people don't realize that a thing that the FDA does not regulate is tattoo ink. And right now, it's estimated that from the age, Americans anyway, from the ages of 18 to 35, about 25% of that population has at least one tattoo. And so one issue that we found recently is microbial contamination. And we're looking at different tattoo inks that we get from vendors, and you can see that of the 85 unopened sealed tattoo inks that we looked at from 13 different countries, nearly half were contaminated, with microorganisms that some species that are associated with skin infections. And so, of course, that's where these are going, right underneath your skin. And so we're following up with studies to see also if there's endotoxin contamination, and we're looking at heavy metals as well in tattoo inks. Here's a couple of different publications that we have put out there now on these microbial contaminants and tattoo inks. Imaging, we really ramped up our imaging and our bioimaging area, capabilities in the last few years. We have all the in vivo imaging capabilities that you would find in a clinical setting. So your MRIs, your CTs, your PETs, we have all that available that we can do in our animal population there. You can see just some of the capabilities that we have there. We actually have this small bore 7 Tesla MRI that we can use, and then we also have a larger bore 4.2 Tesla now that we've recently installed at the center. And some of the things that we want to do with this is, again, this is a non-invasive way that we can look for these biomarkers of neurotoxicity. So instead of using the old classical histopathology approaches, we can do this, with non-invasive techniques. And so we wanted to look at MRI and see if it could inform or direct the plane or the loci of brain tissue slices that are taken for classical neuropathology. So that's a very labor-intensive thing. If you think about looking at a rat per se, and that you want to see what areas of the brain that are affected by a particular toxin, you have to take that brain out, you have to section it into multiple slices, and then histopathologist has to go through that, that tedious brain slice one by one and look for that pathology. So we wanted to see if MRI could be used to visualize the whole brain and provide morphological information. And so we looked at T2 mapping to identify edema and bulk cellular ion pump activity. And so this was our proof of concept there. And you can see just on the right there, that's your classical biomarkers of neurotoxicity. So T2 is actually transverse relaxation time. So that's your spin. So you're looking at increased water content. So that equates to increased T2 reaction times. So, you're looking at hydrogen here. And so your top row is controls. And then your second row is the same animals 48 hours after a canic acid treatment, which is a known neurotoxin. And you can obviously see changes there in the T2 mapping. And when we went and looked at this using the classical histological stains over here in those areas, we saw the same thing. So basically the bottom line here was we actually proved that we could use T2 mapping to give us the same information that we would get from the classical histological staining. Gadolinium, of course, is a contrast agent that they use in MRI. And so what we wanted to do was compare IV administration, which is what is typically done in the clinic, to IP administration to see if we saw a similar effect. And we were indeed able to find that using this OmniScan, which is gadolinium, when we used IP instead of an IV administration, we actually saw the same thing. So T2 decreased proportionally to the number of doses, and at the end there is a significant difference between control and the IV versus IP, but there's no difference between IV and IP. So the T2 MRI can detect gadolinium accumulation, and the route of administration really is irrelevant. MALDI imaging is really a fascinating new area where you can use, MALDI mass spec to do organ imaging. And you can see here that we are looking at doxorubicin, which is used to treat heart issues. And so you can see in the mouse heart there, endogenous lipids there in the rodent brain, and also endogenous lipids in a whole zebrafish. And we have zebrafish capability as well. And all of this could be detected. And so, what's just fascinating to me about this is that when you label, your compound of interest, you can look at that organ and find out exactly what regions that it's winding up in a particular organ in a particular species, after it's administered to the animal. Bioinformatics and biostatistics, of course, this is a huge area and it's growing with the big data. And we're involved with a lot of software development, database development, and helping our colleagues at headquarters looking just at the massive amount of data and trying to help the reviewers, in the process when they're trying to look at the data from a new drug that's been submitted. to see if there's enough data that they can make an informed decision there. We currently have 4 branches in our division of bioinformatics and biostatistics. We call it DBB, but the bioinformatics, biostatistics, and also scientific computing, and what we call research to review and return, the R2R. And current staff, we have about 50 FTEs in this group. They do about 40% of just basic research, but then they also provide support service not only to NCTR, but to the product centers. We have a liver toxicity knowledge base that we developed. We have an endocrine disruptor knowledge base that we developed. We're participating in our Tox 21 forecast, our ToxCast project. So these things that you may see out there in the news that the FDA is working on, we're actively participating in those. Precision medicine, of course, is a big area that we've been working on for several years now, rare disease and drug repurposing. And drug repurposing kind of came to really kind of developed a life of its own when the COVID pandemic started, because we were looking at drugs that had already been approved to see if they possibly could be repurposed. And so we have several publications now where we examined a lot of these drugs that were already approved on the market for use in other areas to see if they could possibly be used for the treating of SARS-CoV-2 infection. And so we have some publications out there now that address that. Regulatory application, of course, looking at systems with our Office of Regulatory Affairs, which I call them the police of the agency. These are the guys that look at the products off the shelf that are out there. If you see that there's an ice cream recall or a lettuce recall, these guys are the ones that actually tested these products and found a problem and had to recall the product from the market. We're looking at a artificial intelligence system now to help support the Center for Tobacco Products to look at pre-market review of tobacco products. And then a very successful program that we've developed is called FDA Label. And that's widely used in our Office of New Drugs and our Office of Generic Drugs at CDER to help them review this data that's submitted for drug applications. Artificial intelligence, of course, AI, machine learning, that's big in the news now. And you can see that we not only do text mining, we're looking at AI for FDA documents, we're looking at AI for genomics, but bug safety in food safety or not bug safety, but food safety because of insect contamination is a big issue. And so we're developing in conjunction with Office of Regulatory Affairs that have to go through boxes of cereal or bags of rice, things like that, and look for these different bug parts. And so a lot of people are surprised to learn that entomologists are actually employed by the agency, and they can identify certain bugs by a wing that they find in a product or a leg that they find an insect part. And so we're trying to develop AI for that, where a computer can look at a sample and actually identify what insect may be present in that particular sample by using AI. So just in summary for NCTR, kind of what we're doing, we want to conduct toxicological research that support and anticipate our future needs, try to improve personal, animal, and public health. We still utilize these classical approaches, but we're also heavily moving into these emerging technologies that I've primarily talked about. We want to be a research and technology resource for all of FDA. We are a government entity. We're primarily funded with your tax dollars. We want to do multidisciplinary training, and we do that with not only postdoctoral fellows, but with summer research programs. We typically have about 25 undergraduates a summer that come in from all over the country and work with a mentor, and they work about 10 weeks during the summer and get substantial training. And a lot of them will get their name on publications or scientific abstracts that are presented at international meetings. And of course, we do a lot of collaboration, not only nationally, but globally. Quickly, I want to mention our Perinatal Health Center of Excellence, because this is something that was just established a couple of years ago at NCTR, and this is an FDA effort. We run this out of NCTR, but it's open to scientists across the FDA. And these are studies that specifically address perinatal health. And again, I've talked about, drugs and the importance there of looking at products that are taken in women who are pregnant. And so, we want to look at that and want to focus, because this is an area that was, quite frankly, was neglected for many, many years. And so, it's an understudied population, of course, just for ethical reasons. You don't do typically clinical trials in a pregnant population. But in many of these products are provided to neonates and infants, or they're provided to pregnant mothers, and they've not been studied extensively in these populations. And so we want to address this, and we want to fill these knowledge gaps about safety and efficacy and potential toxicity. So just some of our recently funded studies. We've got 12 funded studies going on right now at NCTR. We've got 10 additional funded studies that are underway at CBER and CEDAR and also CDRH. We're looking again at neurotoxicity of inorganic arsenic. Of course, being in Arkansas, a big rice producing state, actually the #1 rice producing state, arsenic has been in the news lately the last few years. Looking at therapies for Zika virus that we know is a problem in the pregnant population. And again, opioids, tattoo pigments, if that's transferred from mom, to baby. So is that an issue? And of course, these other drug and metabolite screening approaches. So again, this is open to all the centers at the FDA, and they're all listed here. And so we want to look at, we rank these proposals. I actually just finished reviewing several proposals that were submitted for this year's funding. So what's the relevance to the FDA mission? What's the regulatory need? Is it collaborative across these FDA centers? And what's the scientific quality, of course? And the highest priorities given to projects with the potential for regulatory impact, and also changing medical practice or research approaches. And we have, impacted, decisions that have been made now by clinicians by some of the research that's come out of the agency. I just want to thank a lot of the folks that are doing this work and supplied me some of these slides that I showed you this evening. And also, again, want to reiterate, we had our 50th anniversary celebration back on August the 11th. So we are 50 years old now, and we had both a virtual and a in-person celebration. We had the governor spoke virtually, but we had Congressman Westerman came down for the affair, and we also had our FDA commissioner speak, and we signed our new MOU with the state of Arkansas that really is a one of the kind between the agency and an individual state. And so we have the really through that MOU, it really opens us up for collaborative opportunities with all the universities within the state of Arkansas. And Faith, I think I'll go ahead and stop there and open it up for questions.
Any questions? You can either unmute yourself and ask your question directly, or you can place your question in the chat.
I know that was a lot of information in a short amount of time, but I did want to, to give you really a snapshot of all the different areas that we cover within the agency, and also point out again that this work is going on right here 30 miles from Little Rock, and we are open to collaborative opportunities. We're open to summer students, especially me being born and raised in Arkansas, and probably being one of the few PhD level scientists at NCTR that's actually from the state of Arkansas. I always try to go out of my way to promote the state and say that we have the expertise in Arkansas to help out, and we always want to look within instead of going outside. Sometimes I think we have a stigma that's associated with people being from Arkansas, but we want to reach out and collaborate when we can, and promote our institution and promote the state when we can.
any questions? I see a few of you are popping on, and I'm going to be grateful because I'm going to get a picture before long. But any questions for Dr. Patterson?
I do want to mention, Faith, you can relay this to your undergraduate students, especially. Typically, our program, our what we call it, our ORISE Summer Student Research Program, and that usually the applications usually start going out. The announcement goes out typically the 1st of February. And you can get online, you can search, NCTR, you can Google NCTR, you can Google ORISE. But when that comes out, when that announcement comes out, I'll certainly share that with you so you can disseminate that, to your students there. And typically that's a deadline of usually about mid-March for that. And we try to run that program, about Memorial Day through the 1st of August. So it's when, you're out of school. Typically it gives you a week or two, maybe once classes in, and then a week or two before you have to start back in classes in August. And that's a 10-week program. And it's an amazing experience, for our students to come and intern. I've had students over the years that have interned for me there and have gone on to present at international conferences because of the work they've done. I had a student a few years ago from Massachusetts that came and worked for me for 2 summers, and he wanted to go to graduate school and wanted to know where he should go. And I said, well, with what you have on your resume now, he was majoring in biology at the time with a minor, I think, in math. And I said, you can go anywhere you want to go. And again, I think he was trying to sell himself a little short. And by the time, his recruitment was over, I mean, he was getting recruited like he was a 5-star athlete. He was getting flown to UCLA for an interview. He was being brought to UNC Chapel Hill for an interview, to Cornell for an interview. And a lot of it was based on, his summer internships that he had done and what he had on his resume. So you definitely don't want to, take that lightly. And any of those internships are going to greatly add, because that as a researcher, you're getting someone in graduate school that knows which end of a pipette to hold. And I say that kind of jokingly, but you have a lot of undergraduates that start graduate school that don't have that kind of opportunity. And so, really don't know their way around a lab very well. And so when you get a known commodity that's coming in, people really jump at that opportunity to try to recruit, those level students.
So we do have 3 questions in the chat. One is, can university faculty use instrumentation at NCTR, or do they need to be working on a project with someone at NCTR to have access?
Yeah, very, very good question. And, I don't want to, sound kind of, give the old, classic answer, it depends, but it really does depend. A lot of times we do have collaborative work going on. Sometimes it depends on if it's just, if you've got samples, you're going to come in, what you're doing. Right now we're working with UAMS. They had one of their instruments, go down for the count, and it might be a while before it's back up and running. And so, we're trying to help them out, help your neighbor out here, and have them come in. And, but it just depends on how much hand holding really is needed. If it's going to take the researcher a lot of time to train you up because you've never used the instrument, then we're kind of starting with square one. But if you're, familiar with things, it's not going to be a big burden. I've had students come in working on projects, whether it's, they're working on a graduate project, or they're working on maybe even an undergraduate project. We do have a lot of our faculty, I would say I call them faculty, but most of our research have adjunct appointments with UAMS, with UALR, different universities. And so through that avenue, a lot of times students can come in and actually work in our laboratories on projects that would contribute toward maybe their dissertation work or their thesis work or their even an undergraduate, capstone project work. And we can work that out where, where we're getting the benefit, but they're also getting a lot of benefit as well. So, it would have to be, we'd have to look at each situation individually and determine if that can happen. But yes, it does happen. It's not non-existent. I see that the next one here about the tattoo research. You know, that's another thing again, because because it's not regulated right now. We really can't come out and say, oh, avoid this tattoo ink, from this particular vendor, because then we're going to get sued. The FDA is going to get sued. But you really need to be cognizant of the situation that you're in. What really amazes me is when you go in to a particular, say, a tattoo parlor, for a lack of a better word, a lot of times, you don't think about they're actually doing a minor surgical procedure on you. I mean, they're putting a product, into your body under the skin. And, you may walk into a place, and if it looks more sketchy than your dentist office, which you may even have a more minor procedure in a dental facility, where you would walk into a dental office and you say, I'm not getting any dental work done here the way this place looks, but you wouldn't bat an eye to go in and get, maybe a tattoo, in an environment that's much, much worse. So, I would just say you have to be cautious, with that. But again, the microbial contamination is a big issue. Again, we've found about 50% of the inks. And so you've got to be real guarded about a skin infection. And some of the research that we did several years ago, that you can find publications on that. One of the things that we used to have the capability to do at NCTR is look at photosensitivity. And we had the capability to look at how sunlight would affect a tattoo. And you have to remember, like 30, 40 years ago, the only tattoo inks that you found out there were the old India ink. I mean, you had your grandfather or your uncle who was in the military may have come home and he's got an anchor, a boat anchor tattooed on their arm. Everything was India ink. Well, now you see just these brilliant colors. I mean, we've got the yellows and the reds and things like that. And you can do, I mean, it doesn't take much research to go in and look at publications to see that the constituents that make those up, most of them are very similar to automotive body paint to get that type of brilliant color. So you're basically putting car paint, under your skin to get that type of color, which is why we're looking at heavy metal content and those kind of things. But we did find if your skin was compromised, if you had a sunburn or dermabrasion, that we could take a nude mouse and tattoo a nude mouse whose skin is very similar to human. And we could look at them just under regular UV light, and we could see that when we would tag these particles in the tattoo inks, we could see that the tattoo ink was actually moving deeper into the dermis, and it would actually migrate and accumulate in the lymph node. And I don't know about toxicity, but I don't really want anything migrating to my lymph nodes, and maybe causing issues there. So it is something to be cautious about. And I'm really surprised that there hasn't been more push, from a regulatory aspect, into that area, because so many people now, that's just a common practice. Various amount of time for projects. Typically, we cycle our projects in 3 years. When we approve a research protocol, that's typically a 3-year cycle. Now, sometimes we can't get the work done in that amount of time. So maybe in the case of like a pandemic, where all of a sudden you can't get into the lab, and we give an extension to that. But typically, most projects from the start, I mean, I'm talking about, you have an approved protocol and you walk in the lab, to when you close that project out, where you've got publications now that are coming out. The wet lab work, the animal work may be just a matter of months, but we may generate so much data that it takes a while to analyze all that and get that validated and get the publication out there. What I didn't point out is our center typically publishes about 150 peer-reviewed manuscripts per year. That's That's just on average. How many people are on one project? Again, it can be one of these multidisciplinary projects. We may have people on the bioinformatics, biostatistics side helping out. It may cross divisions depending on what type of project it is. It can be as few as 3 or 4 to as many as 20 or 25. It just depends on the complexity of the study and how many things we want to look at. You may be looking at a genomic aspect over on the cancer side of things, but somebody in neurotox wants to look at a brain effect. So we try to get as much information out of one study as we possibly can. Yes, mostly master's, PhD in the research and development space. We have about 150 PhD level on campus, some master's levels. But that's typically, but again, the support scientist and the again, postdoctoral fellows, we do a lot of training in that space. Yes, we look for expertise on particular projects. We know because it's just like anywhere else, people tend to work in silos unless they know that someone has a particular expertise, and that's where I come in. I oversee the Office of Research, and so if somebody comes to me and says, I want to do this project, and I say, well, this looks like it would be great for this person to work on because they have expertise and you don't have to go outside, or maybe you can go to another center within the agency. We had a project recently where someone was wanted to look at aplesia, I think. And we found out we had an expert at CFSAN that did this type of work. And so we put them in touch with this person that could help them out on the project. And so, yeah, across the agency, we have a lot of expertise, and that's really my job to know what's going on at the other centers as well. We've done a lot of work with UAPB in the past, but not as much with their aquaculture because we typically don't get into the whole food aspect when it comes to that. That's primarily the USDA that deals with that. It's kind of a gray area sometimes when it comes to who regulates what in the food area, but UAPB does a lot in the food animal type space, especially with their aquaculture area. We use zebrafish primarily because they are a good high throughput screen and they're translucent. And so when we're looking at the nervous system on a zebrafish, we can put them under a light microscope and we can see their whole notochord area and to see if there was any effects of a particular agent. And they're really easy to dose because you just put the agent in the water. So we have, and they're a tropical fish. They develop really quickly, and we can look at the embryo and the embryo development with zebrafish. But we have exchanged a lot of information with our different aquaculture facilities because of their expertise over there.
Okay, any other questions? That is all that I saw in the chat.
And again, you've got my contact information, Faith. I mean, if people want to reach out, I'm always open to get an email, and we'll respond. That's primarily what I do all day is sit at a computer now and look at projects and look at emails.